on the slides. So as mentioned, I'm at Imperial, but only since the 1st of September. So I still have my Cambridge logo on the slides, also because most of my group is still in Cambridge. And transitioning over this year to Imperial, and obviously most of the results, if not all of the results I will be presenting today, have been obtained through my work in Cambridge. So we are interested in antibodies in particular. We do protein design more broadly, but I would say most of our focus is on antibody. And the reason for that is that antibodies are really key tools in research and diagnostics, and they are sort of blockbuster drugs. This is a rather outdated, it stops in 2020, plot of the number of approved drugs. I think now we are almost doubling this, so the trend has been continuously, it's been looking exponential. And the other exciting thing is that up to this date, it was mostly monoclonal antibodies like this one, and now there are a lot of exciting new modalities for biologics, like multispecific, bispecific, T-cell engager. There is a zoo of this type of molecule. So a boring slide to make sure everyone is on the same page with sort of the jargon of the field and the structural biology. So an antibody, as produced by our immune systems, is an IgG molecule, which is a Y-shaped protein, is symmetric, is tetrameric, it has two identical heavy chains, two identical light chains. This part at the bottom of the Y is the Fc domain, is, I would say, rather well understood, and its function is to interface with the immune system in various ways, as well as lysosome escape. While the binding to the pathogens or to the target happens in the paratope region, which is by definition the binding region of the antibody, which is at the tip of the Y on this VH and VL domain, and is contained in 6 hypervariable loops, which you can see more clearly in this representation, that are called complementarity determining regions, or CDR. So I will say CDR quite a lot. That's what I mean. And we are interested in studying this part because this part is the most variable part, is what makes each antibody unique and able to recognize a different target. And we work a bit with the full IgG monoclonal, also quite a lot with other fragments, like this guy is called single chain FV, is just the VH and VL connected with a linker, is a monovalent binder. And this one is called a nanobody, or VHH, which is just the VH domain in isolation. And this was originally discovered in some camelid species. Essentially, is an antibody that can fold and function independently of a light chain. And this one only has 3 CDRs, while the other would have 3 per chain. So this was a sort of jargon. With that out of the way, if you do want to obtain a new antibody for a target of interest, nowadays you can use 2 broad classes of technology, which I grouped into in vivo methods like animal immunization, or in vitro methods where you build these large libraries and you screen them for the binders to the target you are interested in with things like phage display or other display techniques. These have been widely successful. But they remain relatively costly and long to deploy, especially the in vivo route. And more excitingly, there are still quite a number of unsolved challenges. Here I list perhaps the 3 main ones. One is that it's very hard to use these approaches to target an epitope of choice. An epitope is the binding region of an antibody, so it's hard to obtain the specificity to a particular region of your target. There are still some antigens or targets that are very hard, like unstable protein, protein aggregates, some other membrane receptors, and so on. And it's very hard to do what I have called here multi-trait optimization, by which I mean finding antibodies that not only are very good at binding, but also have a good balance of other key properties like stability, solubility, and so on. So let's say this region of space here is the gap, the knowledge gap we are trying to fill with our work. And the way we are trying to do it is by introducing a new generation of technologies based on computational design in silico. And we think we can do this now because we have these very powerful computers and algorithms like AI, but much more importantly, we have a good number of high-quality structural data and a huge number of antibody sequences from NGS of repertoires. And so we think that this paradigm shift will enable the development of this class of technology, exactly like it happened in the past for the first and second generation of technologies of antibody discovery. So I'd like, in the next 40 minutes or so, to share 3 stories with you. Our effort in the computational design of nanobodies to target predetermined epitope, a story on nativeness and humanization, which will become clear when I introduce it, and finally, a rather brief story on developability, and in particular on this multi-trait optimization. So in one of my earliest contributions, let's say, to the field when I was still young and not an independent scientist, was to work on the design of antibodies targeting linear epitopes, so linear fragments within amyloidogenic antigen. Amyloidogenic antigens are proteins, typically human proteins, that are involved in so-called misfolding diseases. The most famous of these are Alzheimer and Parkinson, where the protein aggregates and this for this long amyloid. At the time, I was working in labs that were interested in this, and so these examples are in vitro binding data against a protein called alpha-synuclein, which is a protein that aggregates in Parkinson. When we design a single binding loop, like in this one, we get this binding affinity in the mid-micromolar range, which are not that exciting. I mean, they are from a computational design perspective, but not for use. They are not really useful. But when we start to design 2 loops that should bind synergistically, a bit like natural nanobodies would do, then we get affinities that are much more reasonable. We have also been able to test the biological activity of these objects in very simple model of alpha-synuclein mediated dysfunction. So these are worms. They overexpress human alpha-synuclein. The protein aggregates when the worm ages, and so after a few days they are all paralyzed and live shortly. But if we treat them with the design antibody, then we recover the phenotype of the wild type. I mean, in reality, obviously there were a lot more controls, but this is just to show a fancy video of sad worms and happy worms side by side. In more recent years, we have been able to use the same approach to go against a class of traditionally hard targets for traditional antibody discovery. The hard targets in question are these oligomers. So in the aggregation pathway, a very simplified version of it, you start from a monomeric functional protein like synuclein or amyloid beta. In this example is amyloid beta that aggregates in Alzheimer. And then eventually you form this very big, very ordered amyloid fibril that are the hallmark of these diseases. In between, you populate these fleeting intermediates that are called oligomers, which are very toxic, but also very short-lived in the sense that they either convert to fibril or dissociate back to monomer, and they're also very structural heterogeneous. So this is what makes them a hard target. And in this paper, which was some computational design that I did and a lot of experiments done by Francesco Aprile, who's a professor at the chemistry department in Imperial now, we were able to isolate one nanobody that binds very preferentially to these oligomers as opposed to monomers or fibrils. And again, this particular approach, which dates to 2015, has now been used in a broad number of collaborations and papers and so on. In more recent years, and this is work from my research group, we have sort of generalized this idea to be able to design nanobodies that target any sort of structured epitope, so proteins from which we know the structure of or where we can model with things like AlphaFold structure. So the video that played was the spike protein of SARS-CoV-2, so I don't think I need to introduce that, but it was the very initial, very low resolution cryo-EM that came out, at the beginning of the pandemic. And this one in red is the ACE2 binding region of the RBD, which was well known even back then. And so it's a non-neutralizing epitope in the sense if you bind something here, it cannot engage with ACE2. And this blue thing is a CDR3 loop that we designed with our approach, and then we grafted it into a nanobody. So when we went to the lab, we got this binding affinity in the high nanomolar range. We always use negative controls, which are the exact same nanobody scaffold with a different CDR3 loop. In this case, that was designed to bind trypsin, so not designed to bind RBD, and it doesn't bind. We also confer the binding in solution with another, similar negative control. We get comparable KD. This is just the receptor binding domain. This is the full spike. And we have also observed a competition with ACE2, which make us confident that we are indeed engaging this epitope, an epitope in this region. It's sort of indirect evidence for the binding epitope. And again, in reality, we did 6 nanobodies and 3 targets, and I'm just going to show you another target, which is human serum albumin. It's interesting in the field of nanobody because nanobodies per se have a very short half-life, but if you conjugate them to an albumin binder, then they get a very long half-life comparable to that of traditional antibodies. And here we did 3 designs, 2 that targets this overlapping epitope and 1 on the other side. And again, we see this high nanomolar KD, and we confer the binding. Interestingly, in this case, we designed 2 loops. What we were hoping to see, of course, was that we would get a much better KD, but it's not really the case. We still get KDs that are in the same ballpark as the one with the CDR3. I don't have the experiment here, but it's in the paper. We have also conferred the competition pattern. So these 2 compete with each other for binding, as you expect from the epitope, and they do not compete with this other one.
So yeah, you mentioned initially the hard part about this was that conformation changes from time to time, right?
Which conformation?
That the conformation of the antigens changes from time to time, like that they are oligomers that are sometimes...
No, sorry, this was a different story. So this protein is human serum albumin, is a very stable protein. It doesn't change. So this is a different method. So that was just a snapshot of an old paper from 2020 where we have nanobodies that bind this oligomer. But this one is a very stable protein. And again, we also measure the stability of these nanobodies, and this is a plot of the thermostability of natural immune system-derived nanobodies. These are our design, and we are in the, when we design 2 loops, we are sort of in the range. Where we design a single one, we are in the very high part of the stability distribution. So to wrap up this first story, we have shown through various applications that we can design de novo nanobodies to target some predetermined epitopes. Some because these are fragment-based approaches. I didn't go into the technicalities of the computational, but in some cases we have epitopes for which we don't have good fragments, and so we cannot design them. The KD, so the binding affinities we do get, are excellent for de novo design, but are very far from what's a therapeutically useful range. And then the negative controls we have employed throughout demonstrate that the binding is selective, but it's also very likely that the actual specificity or the degree of stickiness of these artificial nanobodies is not enough for diagnostic or clinical application. And then, of course, these sequences we design are very artificial, and so they are likely immunogenic. So these are the sort of things we try to overcome with the currently ongoing research. But if we look at the problem more broadly, so we forget about the specific challenges for computational design, but we just look at the challenges for antibody development in a broader sense. These are sort of the challenges. So on the functionality side, which means the biological activity you want your antibodies to have, you need to bind, as I say, to functionally relevant epitope. In some cases, you need something called species cross-reactivity to enable your tox studies, which means you want an antibody that binds the human protein, but also the mouse protein and also the monkey protein, because in that way you can generate the data you need to start clinical trial. But the main challenge is perhaps this one. is to obtain the desired biological activity of what is ultimately a relatively low dose and low administration frequency. And the consequence of this is that the binding affinity of therapeutic antibody is typically in the tail of the affinity distribution of natural antibodies. So it's typically single-digit nanomolar or better, so picomolar range. And then on the developability front, you have a whole range of other challenges. So the developability means the likelihood of taking an antibody or a drug candidate with suitable functionality and turning it into a safe, manufacturable, and stable product that can be formulated to high concentration and retain a long product shelf life. And the challenge here is fundamental is that we are asking our recombinant protein, our products, to survive a lot of stresses that they didn't evolve to endure. So if we look at natural antibodies, the total IgG concentration in the serum is around 10 to 20 milligram ml, and the half-life in our body is around 20 days. But a therapeutic antibody must be formulated if you want to go for subcutaneous delivery, which is the most convenient from the point of view of the patient. It needs to be at least in the 50 to up to 200 milligram ml, and this is a single monoclonal, it's not a mixture of IgG. And the shelf life needs to be at least 1 year. And these numbers sort of put a strong pressure on this molecule, and the challenges there are aggregation and unfolding, are viscosity when you want to inject, and also chemical degradation. So the bottom line really of these facts is that to take an antibody and turn it into a therapeutic, what you usually need to do is push most of the biophysical properties beyond the typical natural level, beyond the typical average. So either you do it by engineering or you do it by screening a lot of antibodies until you find that one sort of needle in the haystack that tick all these boxes. And these are the biophysical properties, or some of the biophysical properties that are important. I would say they are roughly grouped into those that underpin developability, like colloidal stability, conformational stability, which is the stability of the native state, solubility, viscosity, in part also pharmacokinetics, and then functionality is obviously affinity, but also specificity is very important. And you can also tune the effector function, so the SC region, and you may have other requirement of functionality if you want to get fancy, you want to have a pH-dependent binding to target a tumor, or to get sweeping antibodies or multispecificity and so on. And then, of course, there is safety and immunogenicity. So these are not only a lot of things to monitor, but they are also closely intertwined, by which I mean is if I do a mutation that improves affinity, very often is going to impact the other properties, and unfortunately, very often is going to impact them in a negative way. So for example, if I want to boost stability, I can increase the hydrophobicity, then the native state will be more stable, typically, but this will compromise solubility, so they will also like to aggregate more. The metaphor we use is that of a rubric cube, whereby it's relatively easy to solve one face of the cube. So I can do phage display. It's well established. You can do it to push the affinity beyond the natural level. It's not that hard nowadays, but this usually comes at the expenses of other things. So solving the full cube is really much harder. And in fact, a well-known fact is that the hits that have the best functionality are typically not the one that have the best developability. Yes?
Which of those do you have the lowest throughput assay?
The lowest.
Which of those?
Experimentally, hard to tell, but I mean, always is the in vivo, right? So these are by definition the lowest. Well, safety, pharmacokinetics, also specificity is tricky. There are tons of assay that probe specificity, but none of them is actually probing the real thing, the thing that matters, which is the one in the body of the patients.
That's some of the immunogenicity requirements now, haven't they?
Yeah, it's a bit of an area that is evolving because immunogenicity is a two-sided problem, if you want. So on one end, there is a safety consideration. And I think on that side, they have relaxed it a bit because in reality, it's rare, it's possible, but it's rare that a protein that is immunogenic makes you sick or unwell. So that part has been relaxed. But the other problem with immunogenicity is that your half-life will go down. So if you have an anti-drug antibody response, it may not be toxic for the patients, but it's going to hamper the functionality of your drug because it is going to be cleared much faster. So even if the bar to show safety has moved, it remains a major problem because then you cannot show efficacy. So there is this. So the way we are sort of trying in our group to address the complexity of this problem is by developing what I call a toolkit for computational antibody design. So in this blue shading, and I obviously will not go through all of this in detail, but in this blue shading are our methods for binding design or antibody discovery. I've presented to you, I've shown you these results before. So this is our fragment-based approach that we have used for the spike, for serum albumin. We are now using a lot more AI, both autoregressive AI, so MPNN and graph neural network, things like that, and diffusion framework for generative design. But this is pretty much ongoing research. In this pink shading, we have our method that I call for hit selection, because the idea is that you can either design a lot of candidates or obtain by traditional means like a display platform or immunogenicity or immunization campaign. A lot of antibody, and then you need to prioritize them. Obviously, you are going to look at binding, but you also should look at all the developability profile of this molecule. So this is the suite of methods we've been developing to look at that. Here on the x-axis is always our computational prediction. On the y-axis is always one experiment. This particular plot done with Novo is an experiment that looks at self-association propensity or generic stickiness to surfaces. So it's in between specificity and solubility, if you want. This one is really more a solubility measurement. And in both cases, we see that essentially our prediction can replace these rather material-intensive and laborious experiments. We do have outliers sometimes, of course, but mostly we get it right. And this works very well for mutational variants of the same antibody, by which I don't mean a single mutation away. It can be, I think in this particular plot, it was up to 32 mutations. So substantial differences, but it wouldn't work this well if you were comparing completely unrelated proteins. Probably also the experiment wouldn't work that well, but that's a separate issue. This is stability, thermostability to be exact, for nanobodies, and again we have this very good trend. This story is the next story I'll share with you about nativeness. Here is a generalization of our solubility. So now the prediction is the blue line and the experiments are the red marker. And this is solubility as a function of pH, which is a critical parameter for formulation. So we can predict it also rather accurately. And this is an Ig4 human antibody. These methods are what I call lead optimization methods. So these would take a very good or a very bad, it doesn't matter, an antibody or nanobody, in this one on the top, even any protein in reality, and would improve some of their properties by suggesting automatically mutation to do. And I will go in more detail of both. So next story, it really starts from a consideration. The consideration is that if we look at the approved antibody, and these are again all data because nobody has compiled a new data set of this type, but if anything, the percentages are now even more in favor of in vivo. But the bottom line is this in vitro platform, if you read any review on phage display, like the Nobel lecture by Greg Winter, you hear things that are absolutely true, that these are faster, they're cheaper, they afford a much better control over antigen presentation, they work even if your antigen is not immunogenic. It's a very long list of advantages. And yet, people that do drug development overwhelmingly prefer the in vivo route. And now that there are all these animals with a humanized immune system that they make a human antibody even more so than before. So this poses the question of why. And the reason why is in what we call in vivo properties. So in reality, immune system-derived antibodies have a much better balance of these critical properties like specificity, pharmacokinetics, and safety. And these properties obviously are incredibly hard to measure in any good throughput, and therefore are very hard to predict. And to come back to the question asked earlier, it is reported that up to 40% of the biotherapeutics in the market have their activity reduced by immunogenicity, which again, it doesn't mean they are toxic. It means that they are less active than they could be because they get cleared faster by our immune system. So this was a big problem because obviously if we cannot measure them, surely we cannot predict them. So our solution to this was to try to bypass the problem of predicting these properties directly by learning to design antibodies that are literally indistinguishable from immune system-derived antibodies. The way we have done that is to use AI, because we have a ton of sequences from NGS. We have used something called a vector quantized variational autoencoder, but I'm not going to go into the mathematics of this, but fundamentally, when you train this machinery, you feed it a lot of immune system-derived sequences. And what it does through this sort of information bottleneck and other tricks is that it learns the latent distribution of where these native sequences exist. And so when you use it, so at inference time, you can give it any sequence you want, and it's going to measure the distance from this distribution or the likelihood of belonging to the distribution, which is what we call nativeness. So to understand it better, we have trained it on human antibodies. We have also trained it on nanobodies. And we give it human antibodies and get high humanness score. We calibrate the scoring so that 0.8 is a sort of the optimal boundary between native and non-native antibodies. And then when we give it mouse antibodies that it has never seen during training, it's a completely new thing, it knows that they don't live in the same distribution, and so it assigns low humanness score. And so we can use it for things like hit classification and ranking. And this is the exact same thing, it's just more quantitative if you are into area under the curves or things like that. But what it shows is that we can really well separate human antibodies from those of other species. And perhaps even more importantly is this blue control here, which is artificial sequences that we have made in the computer. And the way we have made them is that we have taken the human antibody position-specific scoring matrix, the one in the training set of our model, and we have filled each position of this sequence using the human frequency. So we randomly pick, using the human sequence, each position. So these are fully human if you look at each position individually, except each position has been filled independently of the other. So this very strong, one is complete separation, ability to distinguish them away, tell us that the method has learned high order relationship between different positions in the sequence. So it has learned that this one is human, but it doesn't go with this other one that is human. Yes.
Have some of those sequences tried in the lab and actually been verified as very much not immunogenic?
They haven't been tried, I think, to that extent, but this PSSM approach is how a lot of the synthetic library for phage display have been built. Okay. So certainly they can make antibodies that are folded, that they bind, and so on. Immunogenicity is a big question. We can have a much longer discussion later, but in reality, we know nothing about immunogenicity. That's the bottom line. The other thing this method does, which is very useful for engineering, is that it offers single residue resolution. So it just doesn't give you a score for the whole sequence, but you can also get this profile. This example is a mouse antibody in orange, which is a precursor to a clinically approved antibody that has been experimentally humanized, which is in purple. And so you can see all this region of low nativeness, low humanness in this case, have disappeared, except in the CDR region. So the reason in the CDR are still there is the way these things are humanized, or have been humanized historically, is that they would take the mouse CDR and put them on a human framework. So the other interesting thing is that even if the CDR3 sequence is exactly the same between the two, the profile is different because probably the method predicts that the influence of the other regions that are different is changing the context of the CDR3. And so this suggested we can do design, and in particular, we can do humanization. by looking at this. And we have focused on nanobodies because there are a lot of ways to humanize mouse antibodies and very few to humanize nanobodies, but these are becoming increasingly important in therapeutics. So the way we did it, and I think with the question I'm sort of going very slow, so I'll try to speed up, is that we have this sort of two-way humanization. So we start from a wild type, we try to design mutation rationally. The method suggests them automatically to increase humanness, but we also ask not to compromise too much the nativeness of the nanobody, which is the x-axis here. So we have done 4 different ways of sampling mutation. 2 of them will touch buried positions, so things that are not on the surface, which is not a good idea. And then we compare with this purple one, which is another method that came out when we were working on ours, that does some very sophisticated frequency and structural analysis. And is very effective in our prediction on improving humanness, but it always comes at an increased cost on nanobody nativeness. When we take them to the lab, is a binding assay, what we see, and this is the binding affinity, so here lower is better, it means it binds better on this bar plot. Let's focus just on the bar plot. What we see is that when we touch a mutation position that are in the buried in the native state, we compromise a bit the binding and we retain it otherwise, while the purple one compromises it the most. In here, this was a very challenging case because this is a picomolar binder, so it's a very optimized, they really push the affinity, which means any mutation will impact it. Here we have one design that retains the affinity, another that loses about 1 order of magnitude, but in the purple one, there is absolutely no detectable binding in this concentration range. And when we look at stability, we have a similar outcome, so we tend to increase actually stability among humanization because we have this nativeness control, or we retain it like here in the case of the red, while the purple one either retains it or lose some stability. Again, the story is a bit different when you start to touch a buried mutation, but we don't do that anymore. And so we have used this quite a lot. Here I have a couple of case studies that we did in partnership with companies. This one is CAR-T. So I don't know if everyone knows what a CAR-T is. Essentially, it's a T-cell that we engineer, I mean, people engineer, this company in particular, to display, instead of the normal TCR, T-cell receptor, a nanobody. In this case, it could be an antibody that recognizes the target you want. So when the T-cell gets there, it's going to kill the cell that displays this target. Typically, is in oncology, to kill cancer. This one in particular is for glioblastoma. It targets this particular antigen. So this company had a nanobody derived from a lamb or an alpaca. We did our humanization, the 2 pipeline that I showed before. We retained the binding in vitro, similar story. Then they put them in the CAR, so now is on the T cell and is looking at cancer cell. We look at killing. This assay is always shown relative to the non-transduced T cell, so this is set to 100%, and these are different decreasing concentration of T cell to target cell, and they all kill the cancer very effectively. What's interesting, though, is when they look at off-target killing. So now they're trying to kill cancer cells that don't have this particular target. And what we see is that the wild type has quite a strong off-target killing. I mean, this is the wild type, right? So it still kills a lot of this cell, unlike the non-transduce, while both of our design, and this one in particular, have a substantially reduced off-target killing at concentration that are closer to the in vivo relevant ranges. So when you have a lot fewer T cell than cancer cell, it's non-detectable, the off-target killing. So they took this in vivo together with a lot of other sort of IP from the company. And what they saw is that if you just give the mice with this human tumor in their brain, the non-transduced T cell, the tumor just grows over 14 days as you expect. But if you treat them, the tumor disappears. So this was very interesting. And I'm going to go fast now because I'm running out of time. We have done it with another company in other ways. This is the only difference here is that now we are not just trying to improve humanness with our plot, but we are also trying to improve solubility with our other tools, which is this color code. And we are trying to control stability again with our other tools. So we are doing a more comprehensive approach. But bottom line is our variants alpha and beta retain the TNF alpha inhibition activity at par or even better than wild type. This same company also had things that target a brain receptor, the transferrin receptor, to translocate biologics in the brain. Very similar story. Now the binding is the y-axis in this flow cytometry. It's better to show what didn't work. This particular guy didn't work. It was an attempt to recover something that had lost a lot of nativeness and sort of also a lot of solubility, but this attempt failed. Oh no, sorry. Are we back? Maybe we are. Let's see. Okay. And the other remain the binding. This is the same thing for a different family. They all retain the binding. I just wanted to get to this, which is when they combine them. So the reason to combine them is to see if they can bring the anti-TNF alpha into the brain. So this just shows that the dimer is still active against TNF alpha, as you would expect. These are now fully humanized, one fully engineered by us. And these are data, initial data from a rat model. What they show is in the wild type rat, nothing gets into the brain because they don't bind the transferrin of the rats. But in this humanized rat that has the human transferrin, you get a lot of brain entrance. There are other data that show that without the TFR targeting arm, they don't get into the brain. This is the concentration in the serum. All of this is after 72 hours, so you presumably have lost quite a lot by then. And this is the ratio. So without getting into the detail, the bottom line is that these humanized nanobody were very effective at shuttling into the brain active biologics. So the conclusion here is really that we can design or engineer antibodies and nanobodies that are similar or indistinguishable to immune system derived one, and we can use this approach for screening and for humanization. The final story, I'll try to be quick. Now you only see that I'll be speaking for 1 minute because it crashed before, but the final story is about this, so this multi-trait optimization. So we focus on stability and solubility without affecting or compromising binding. As I mentioned before briefly, these two properties are typically anti-correlated because if you increase charge, you increase solubility, but you decrease stability. If you increase hydrophobicity, usually you get the opposite effect. And they are very important because together they determine colloidal stability, which is the integrity of your formulation, so the aggregation resistant. They correlate with production yield. If you are stable and soluble, typically you produce higher yield, so it's cheaper to produce your drug. They anti-correlate with viscosity often, not always, which is good because you want to inject this thing and you want to have low viscosity. High solubility has been shown to correlate with low polyreactivity, so high specificity, low stickiness around, and high stability correlates with long in vivo half-life, which together means that when you both are high, you should get better pharmacokinetics, and this has been shown for a few data points. And they're also very important to assess new administration routes, in particular oral administration or inhalation, and they are required for some fancy formulation modalities like slow releasing and so on. So our pipeline is, the rubric cubic problem, works by identifying some liabilities for the solubility with our tools, and then we look at conservation. So we have these very curated alignments in this case of other nanobodies. Obviously, we have for human antibodies and mouse antibody and therapeutic antibodies as well, where we look at positions that are not well conserved, and then we predict at these liable sites identified in these two ways, the mutation that are enriched according to our alignment, so that are observed in nature as well, and we score them by solubility improvement on the x-axis, stability improvement, and finally we combine them. The important thing or the interesting thing here is that this step of the alignment is really important because this grounds your algorithm into a space that is evolutionary and biophysically grounded. So you're looking at things that occur in nature. And what we see is that this y-axis in reality is an energy function, is useful computation, is very fast, inexpensive to run, but it's not that accurate. They've been around a long time, they've improved steadily, but never substantially. So roughly 1 in 4 of these prediction. So mutations that are predestabilizing actually are not. But when you ground the state, so when you look only at those that also improve this log likelihood, then you get a lot lower false discovery rate, which is very important because then you can go to the lab much more confidently. And this is what we did. So these are the mutations we selected or combinations. And these plots, the x-axis is now the experimental thermostability that correlates with conformational stability. Here the y-axis, the proxy for solubility, and all of them are improved. Here the y-axis is a hybrid between solubility, but is more cross-interaction. It's more a proxy for specificity. It's a common developability assay. And here, the only one that didn't improve are the mutation to positive charges, because positive surfaces make more cross-interaction, is well known. Everything else improved. And this improvement is really substantial. It's not a small improvement. This is 14 degrees in melting temperature for a nanobody. And 44 seconds in this assay in retention time is essentially the whole dynamic range. The whole dynamic range is probably under 1 minute. And they all retain binding, except, of course, the negative control that is KO here. With Novo Nordisk, I mean, we showed those data to Novo Nordisk. They were interested. So they did a lot more experiment on 2 more nanobodies and 3 antibodies expressed as single-chain fragment. 2 of these are clinically approved antibodies that target TNF-alpha. Adalimumab is also called Humira. It's the top-selling drug of all time. It's a very optimized antibody. It's very hard to push this to get any better. In fact, in the cross-interaction, we didn't make it better because it's already at the low end of the dynamic range. But in the stability, we improve it by up to 9 degrees. And the opposite is almost true for golimumab. Golimumab is here. We improve the stability by a little, but we reduce the cross-interaction a lot. It's actually easier to look at the thing that didn't work, which is only 2 mutations. One that is present in both of these designs for a nanobody. We thought it would make it more soluble, but neither. And one that is this light chain mutation in Humira, which we thought would make it more stable, but didn't. Everything else worked. And so again, we validated this on 6 different antibodies, including 2 clinical ones. And we can automatically design single or multiple mutation. And we have had a success rate in the order of 90%, whereby 39 of 42 of this design had better of equal stability and solubility. And only 2 of these 33 mutations we tried, these are 42 because they combine different mutations together, but there were 33 unique mutations across the 42 design. Only 2 of them were false positive, and only 1 that I didn't show actually affected the binding affinity. And this slide is just showing that we are very excited about antibodies, but a lot of our tools work with protein more generally. So this is an alpha amylase enzyme. It's an enzyme of high industrial relevance. This particular one is from a thermophilic organism, so it's incredibly thermostable. And what we did with our method, we tested 3 models, each comprising 6 or 7 mutations. We retain the hyperstability, which is incredibly hard to even measure because it's super stable. We retain the activity, maybe we improve it, but I would argue this is in the range of your batch-to-batch variability, but we massively improve the solubility. So this is a 9% improvement in this assay, which is a really substantial improvement in PEG precipitation. And to conclude, we have a web server where people can use our method, and people need to register with an email. So about 2/3 of the 1,500 active users in the last academic year use an institutional email to register. And then I can use the extension of their institutional email to make this fancy word map. And this is essentially where they work. So it was very humbling to see that this method has been taken up by the academic community, I would say almost worldwide. Or at least being used once. So with that, I'd like to thank the crazy people who give me the money down here, but more importantly, the people who did the work, which are the members of my group, in particular Ruben, who's the only person in Imperial with me at the moment who did all the humanization part, and then collaborators in various companies that we work with. And I'll just leave you with this sort of visual summary of the 3 stories I shared.